Dear students, happy to meet you in this online mode of education. Today we are going to discuss about the topic of gene mapping. It is coming under the paper genetics. I am Dr.S.ANGELINA PARIMALA, Associate Professor of Zoology and Head PG and Research Department of Zoology, A.D.M. College for Women, Nagapattinam. Let us see something about the introductory part of gene mapping. Let us see the definition of gene mapping. Gene mapping, this refers to the techniques used to identify a gene's location and distance between genes. The distances between various sites inside a gene can also be described through the gene mapping. Placing several molecular markers at specific locations on the genome is the fundamental element of all genome mapping. There are many types of molecular markers. When creating genome maps, genes can be observed as a particular class of genetic markers mapped similarly to other markers. A genetic map, which is otherwise called as recombination map, is a representation of the linear order of genes, that is, in the loci, and their relative distance determined by crossing over frequency and along the chromosomes. And also, in fact, we can say that such linear maps can be constructed, supports the concept of genes being arranged in a fixed linear order along a single duplex of DNA for each chromosome. We can use the recombination frequencies to produce genetic maps of all the loci along each chromosome and ultimately in the whole genome. How we can calculate the distance in the mapping? A genetic map shows the relative location of two or more genetic traits. Usually, we analyze the offspring in a particular cross and track how many times two given genetic traits are inherited together. For instance, eye color and wing shape of Drosophila. The higher the percentage of progeny that inherit both traits together, the closer the genes responsible for the traits on the chromosome. When we see the gene mapping, here the process of locating genes within a genome is very important. The regions of a genome that directly encode proteins or the protein coding genes are usually of great interest to scientists. Therefore, it is often a priority to locate each gene in genome. And also, nowadays, a mapping procedure typically involves genome sequencing and analysis of the resulting sequence using digital methods that would let us spot desired genes. And also here most of the gene mapping projects begin with genome sequencing. Let us see something about the types of gene mapping. When we see the types here, the genetic linkage maps and physical maps are of the two main categories under the gene mapping. And also both maps consist of genetic markers and gene loci, while physical maps involve actual physical distance, often measured in number of base pairs. Distances of genetic maps are based on genetic linkage information. There are many gene mapping methods, including comparative, physical, and genetic linkage mapping. However, the physical and genetic linkage mapping are very, very common. Let us see something about the linkage mapping here. It is based on the recombination frequency between genes. It provides the relative positions of genes on the chromosome. When we see the physical mapping, it determines the actual physical distance between the genes or markers on a chromosome measured in base pairs. When we see comparative mapping, it compares the genomes of different species to understand evolutionary relationships and identify conserved genetic regions. Let us see something about genetic linkage mapping. Here, this genetic linkage map shows the location of each gene on a chromosome and their relative distance from one another. Initially, these maps were created by tracing the inheritance of several features like eye color and hair color, blood, saliva, or tissue samples from both affected and unaffected family members are used to begin a genetic map. Saliva is the most common, often used sample in genetic mapping, particularly personal genetic studies. When we see this mapping is made possible by crossing over a regular biological occurrence during meiosis, that is, during the cell division that produces sperm and egg cells. Here the chromosomes line up in pairs in the center of a cell during the first phase of meiosis, where they often stick to one another and they exchange similar fragments of themselves, also known as crossing over. And here it makes it possible to determine which gene is present on each chromosome and where it is located within that specific chromosome. Based on the distance between two genes, mapping can also determine which gene is more likely to undergo recombination. When we see the physical mapping here, it always provides the actual DNA base pair distances between landmarks. It is one gene mapping approach that has a high degree of accuracy in determining the sequence of DNA base pairs. A physical map provides the nucleotide numbers and the precise physical distance between genetic markers. Radiation, hybrid mapping, sequence mapping, and cytogenetic mapping are the techniques used to produce a physical map. Here it assembles larger DNA sections using DNA markers and DNA fragments. Research shows that to identify the locations of the DNA bases from the overlapping sections of the fragments. Various physical mapping methods are available to study genomes of multiple sizes and obtain different degrees of precision. Physical mapping is the common approach used in genome sequencing to acquire an entire genome sequence and determine whether there is any correlation between the specific DNA sequence and phenotypic features. Let us see in detail about the techniques in gene mapping. Let us see the first one, that is linkage analysis. Here the principle behind this is genes that are close to each other on a chromosome tend to be inherited together. The frequency of recombination between them indicates their distance. When we see the tools here, we are using genetic markers, for example, SNPs, microsatellites, and then pedigree analysis. When we see the process by analyzing the inheritance patterns in families, researchers calculate recombination frequencies and construct linkage maps. Second one is restriction fragment length polymerization. You can say polymorphism. Here the principle behind this, variations in DNA sequences can create or eliminate restriction enzyme sites, leading to different fragment lengths. When we see the tools, the restriction enzymes, gel electrophoresis, and DNA probes. When we see the process, DNA is cut with the restriction enzymes and fragments are separated by gel electrophoresis. Probes detect specific fragments, allowing mapping of genetic markers. Let us see something about fluorescence in situ hybridization. Here the principle behind this, fluorescent probes bind to specific DNA sequence on chromosomes. The tools utilized here are fluorescently labeled DNA probes, then fluorescence microscope. And also when we see the process, yes, the probes hybridize to chromosomes, which are then visualized under a fluorescence microscope, providing physical locations of genes. Let us see something about the radiation hybrid mapping. Here, the principle behind this, radiation-induced chromosome breakage and fusion in hybrid cells. The tools we are using here are hybrid cell lines, PCR, and genetic markers. And also the process includes cells are irradiated, causing chromosome breaks. Hybrids are analyzed to determine the presence of genetic markers, constructing high-resolution maps. And also when we see the genome-wide association studies, here the principle identifies associations between genetic variants, usually SNPs are included, and traits or diseases. And tools we are using here are the SNP arrays, large population samples, statistical analysis also we can use here. Then the process includes that compares genetic variants across individuals to identify markers linked to specific traits or diseases. When you see some examples for the gene mapping, number one is about a human genome project. Here the goal for this project is to map and sequence the entire human genome. And also when you see the outcome, here it completed in 2003, providing a comprehensive reference of human genetic material. When we see the second one, cystic fibrosis genetic mapping, here the process includes that linkage analysis identified markers closely linked to the CFTR gene on chromosome 7. The outcome is discovery of the CFTR gene, leading to advances in diagnosis and treatment of cystic fibrosis. And also in the Drosophila melanogaster, here the process includes linkage maps constructed using recombination frequencies between visible markers. And also when we see the outcome, here the detailed genetic maps that have been crucial for understanding developmental biology and genetics. When we see in the field of evolutionary biology, here we can take the example. Comparative mapping helps trace evolutionary relationships and the conversation of genetic regions across species. When we see in the personalized medicine, here the example includes identifying genetic variants that affect drug metabolism and response, enabling tailored treatments for individuals. When we see the sequencing of the genome, here sometimes mistakenly referred to as it is a genetic mapping by non-biologists, the process of shotgun sequencing resembles the process of physical mapping. It shatters the genome into small fragments, characterizes each fragment, then puts them back together. More recent sequencing technologies are drastically different. While the scope, purpose, and process are totally different, the genome assembly can be viewed as the ultimate form of physical map, in that it provides in a much better way all the information that a traditional physical map can offer. And also mapping mutational sites. When we see the mutational sites related to mapping, here in the early 1950s, the prevailing view was that the genes in the chromosome are discrete entities and also individuals by the genetic recombination and arranged like beads on the string. During 1955 to 1959, Benzer performed genetic recombination experiments using mutants of bacteriophage T4. He found that on the basis of recombination tests, the sites of mutation could be mapped in a linear order. These results provided evidence for the key idea that the gene has a linear structure equivalent to the length of the DNA with many sites that can independently mutate. In 1961, Francis Crick, Leslie Bennett, Sidney Berner, and Richard Watts, they performed genetic experiments that demonstrated the basic nature of the genetic code for proteins. These experiments involving mapping of mutational sites with the gene of bacteriophage T4 demonstrated that three sequential nucleobases of the gene, the DNA specify each successive amino acids of its encoded protein. That the genetic code was shown to be a triplet code where each triplet specifies a particular amino acid. They also obtained evidence that the codons do not overlap with each other in the DNA sequence encoding a protein, and that such a sequence is read from a fixed starting point. And also scientists Edgar et al. Performed mapping experiments with all mutants of bacteriophage T4, showing that recombination frequencies between mutants are not strictly additive. The recombination frequency from a cross of two mutants is usually less than the sum of recombination frequencies for adjacent internal subintervals. Although not strictly additive, A systematic relationship was demonstrated that likely reflects the underlying molecular mechanism of genetic recombination. When we see the importance or the significance of genetic mapping or chromosome mapping here, we can identify the disease genes. It helps to locate genes associated with genetic disorder. For example, we can easily identify the breast cancer, and also it facilitates the development of diagnostic tests and targeted therapies. When we see here again, it helps to understand the genetic disorders, provides insights into the genetic basis of diseases such as cystic fibrosis and Huntington's diseases. And also in the agricultural improvements, when we see here, it helps to identify genes related to traits like disease resistance, yield, and quality in crops and livestock. And also it helps to assist in breeding programs to enhance desirable traits. When we see in the field of evolutionary biology, it helps to compare mapping may be made across species, reveals conserved genetic regions and evolutionary relationships. And also in the personalized medicine, maps helps to see the genetic variants that affect drug response, leading to more effective and personalized treatments. When we see the challenges and the future directions here, the first challenge is complex traits. Mapping genes for traits influenced by multiple genes and environmental factors remains challenging. And also in the technological advances here, the next generation sequencing and advanced bioinformatics tools are improving the resolution and accuracy of chromosome maps. And also in the related to ethical considerations, as chromosome mapping reveals more about genetic predispositions, ethical issues regarding privacy, discrimination, and consent must be addressed. And you see here, we can see some examples. For identifying disease genes here, we can give the example. It is crucial for understanding breast ovarian cancer risk, and also in the understanding genetic disorders. Here we can give the mainly muscular dystrophy. We can give an genetic disorder, and also in the agriculture improvements here, the mapping genes related to disease resistance, yield, quality traits, especially in the crops and livestock. And also the chromosome mapping is a fundamental aspect of genetics that provides detailed information about the location of genes and genetic markers on chromosomes. This information is crucial for understanding the genetic basis of traits and diseases, improving agriculture practices, and advancing personalized medicine. Through a combination of linkage, physical, and cytogenetic mapping techniques, researchers can construct comprehensive maps that enhance our understanding of the genome and its functions. When we see the benefit of mapping in the human genome here, the sequencing offers a molecular diagnostic that enables physicians to precisely customize treatments that can significantly improve the possibility of survival. Specific medicines targeting particular gene mutations can replace trial and error medicine for several disorders. When we see the DNA mapping here, it refers to the range of techniques available for describing the location of genes. Similar to that, the topological maps of a city or a country, DNA maps can display various levels of detail to show how far two genes are from one another. And also, the recombination events are used in genetic mapping techniques to measure the distance between genetic markers. And also random fragment length polymorphism. Here it measures the difference in homologous DNA sequences to calculate the distance between two markers. And also currently the gene mapping analysis targeting single gene disorders use short random repeat polymorphism. And also related to single nucleotide polymorphism, here it is used in genome-wide association and linkage analysis in the genetic research. And also linkage analysis is studied using the inheritance of characteristics and genetic signatures like SNPs and microsatellites, and also Genome-Wide Association (GWA) that studies the connections between traits and markers like SNPs and microsatellites by treating the population as a single family. The method is used to map the gene functions of common disorders. When we see this one is related to disease association here, the process to identify a genetic element that is responsible for a disease is also referred to as mapping. If the locus in which the search is performed is already considerably constrained, the search is called the fine mapping of a gene. This information is derived from the investigation of disease manifestations in large families, that is in the genetic linkage group, or from population-based genetic association studies. Using the methods mentioned above, the researchers are capable of mapping disease genes, and also generating a gene map is a crucial first step towards identifying disease genes. And also gene maps allows for variant alleles to be identified and allow for researchers to make predictions about the genes they think are causing the mutant phenotype. An example of the disorder that was identified by linkage analysis is cystic fibrosis. Here, the samples from 50 families affected by cystic fibrosis were analyzed using linkage analysis. And also, hundreds of markers pertaining to cystic fibrosis were analyzed throughout the genome until cystic fibrosis were identified on the long arm of chromosome 7. Researchers then had completed linkage analysis on additional DNA markers within chromosome 7 to identify an even more precise location of the CF gene. When we see the challenges and the future directions here, first is about the complex traits. The mapping genes for complex traits influenced by multiple genes and environmental factors remains challenging. Technological advances here, the advancement in sequencing technologies, bioinformatics, and computational methods are improving the resolution and accuracy of gene maps, and also ethical consideration as gene mapping reveals more about genetic predispositions. Ethical issues regarding privacy, discrimination, and consent must be addressed. And also gene mapping is important for the process of determining the specific locations of genes on chromosomes. This process helps scientists to understand the genetic architecture of organisms and is crucial for identifying genes associated with diseases, traits, and evolutionary relationships. It is the cornerstone of modern genetics, providing insights into the structure, function, and evolution of genomes. By pinpointing the locations of genes, researchers can unravel the genetic basis of traits and diseases, paving the way for advancements in medicine, agriculture, and understanding of our life itself. When we conclude, this gene mapping refers to the process of determining the location of genes on chromosomes. Today, the most efficient approach for gene mapping involves sequencing a genome and then using computer programs to analyze the sequence to identify the location of genes. Thank you.